Well, hello everyone. During this speech, we will talk about the project we have been working on during CEMRACS. It's about multiscale population dynamics, interaction between scaling, developmental, and reproductive biology. I worked with Céline during this project, and well, we quite enjoy working together. So for the outlines of our speech, they are given by that. We will talk about some biological backgrounds on ovarian follicles. We will present some modeling approaches that we did to follow this process. And we will talk about some theoretical and numerical results that we could have for our project. Well, okay, we will present a model to follow the growth of follicles. So what's follicle? Follicle is a structure that is composed from egg cells that lives in the ovary of a female. And that is, the initial stages of these follicles are the quiescent follicle. Once these follicles are activated, and this activation can occur at any time, these follicles go through several successive growth stages. Starting from the initial stage that corresponds to the quiescent follicles, and we can see that during these successive development stages, the follicle changes its structure, and we can see that there is an increase in the size of the follicle during the development stages. We should know that at a given time in the ovary we have the coexistence of several follicles belonging to several development stages in the same time. Well, that allow us to classify those follicles by their size to make them into different compartments according to the size of follicle. And so we can follow the number of these follicles, the density of this follicle on the population of follicles. And we can also identify the parameters or the set of parameters that are influencing the dynamic of this process. So here we are interested about two sets of parameters that are the lambda, the maturity rate, and the mu, that are the death rate of follicles in each compartment. And the compartment here are representing different maturity stages of the follicle. So, from a statistical point of view, we have here on the left a figure that gives the quiescent follicle pole development or progression according to the time. We can see that this curve is divided on two main parts that are two straight lines, and we can see that the regression factor on the second curve is more important than the one in the first part of the curve. That means that the decrease in the number of follicles is higher on the final stages than on the first stages. And we can also notice the fact that the pool of quiescent follicles goes to the exhaustion. So at a certain time we have a very, very small number of follicles that are still on the ovary. On the figure on the right, we have the distribution of follicles according to the size for three different species. For the female, human female, sheep, and rat. Well, we can see that those distributions are quite similar for the three species. We have this profile with two bumps. And we have to highlight two important points, that we have the preservation of this profile according to the age. So we can see, for example, for the human female, we have the same profile in different ages. So we have the preservation of this profile according to the age. And we have also the decrease in the total number of follicles with the age. So when the age is higher, we have a decrease in the number of follicles, which is quite normal. So the question here is, given the biological knowledge, the recent biological knowledge, can we define a non-linear model to modelize and to follow this process that can help us to understand the dynamic of this kind of process in order to be able to follow it or to manage it. And this can have important biological application, for example, in the improvement of fertility for some species, etc. So we'll see. In this slide, we will talk about the regulation. Okay, the regulation is how a sub-part of the population of follicles can influence the growth of the whole pool of the follicles. So we have two important aspects that are coming from biology that we have to incorporate into our model. The first aspect is the negative feedback coming from a part of the follicle pool that is influencing the growth and that is inhibiting the growth of follicles in the initial stages. This is given by the expression of lambda, and here we have this sum that is given a negative feedback on lambda from the population. Well, these expressions are quite common on literature. So this is the first aspect that we can incorporate into our mathematical model by this expression. And the second aspect that we have to incorporate is that competition that get, let's say, higher on the final stages and that allow a very small number of follicles to reach the pre-ovulatory stage. And this is represented by the mortality rate given by this expression. So to fix the idea, we have here some order of magnitude of follicles population for the women. For the quiescent follicle, for example, at puberty, a woman have around between 10 power 4 to 10 power 6 quiescent follicle. And this number is reduced on the menopause to be less than 100, 1,000 to 1,000 follicle. We should also notice that only 400 follicles will ever reach the pre-ovulatory stage. This is due to the huge competition between follicles into the latest stages. So we start from a very important number to reach a very small number at the end. And about the activation of quiescent follicle, we said on the previous slide that this activation can occur at any time. So for the women, we have the activation of only few quiescent follicles per day. Well, here I will leave you with some mathematical hypotheses with Céline to present the audio system.
Thank you. So now you have all the biological background to make some mathematics. So let's go. You understand two things. The first, it was the initial pool of quiescent follicle. It was huge. Okay? So we can introduce some epsilon and make this hypothesis. It was that the initial pool of quiescent follicle, so x0 to t equal to 0, is in the order of 1 on epsilon. And the second thing, it was that the quiescent follicle have a very slow dynamic. So we can make the hypothesis that the two rates, the initiate rate lambda 0 and the death rate mu0 of the quiescent follicle, it's in order of epsilon. Okay. So thanks to this hypothesis, we can make some rescale and obtain a system, a slow, fast system. Okay. In fact, the idea is the first equation here. It's for the dynamic, describes the dynamic of the quiescent follicle. So you have the initiated rate. Okay. So the quiescent follicle go to the next compartment with an initiated rate lambda 0, or die with a rate mu0. So it was this dynamics. And in fact, the rate here, it was not the rate introduced, it was a rescale rate. And thanks to this rescale, we appear some epsilon there. So the second equation describes the dynamics of the growing follicle. Okay, so if we look for the compartment number I, okay, you have some follicle in the compartment before that come with a growth rate lambda I minus 1, or you have in this compartment you can growth and go to the next compartment with this rate, or die with the rate mu I. Okay, so it was right just there. And what is very interesting is this epsilon. In fact, you understand that this epsilon shows that the dynamic of the growing follicle it's faster than the dynamic of the quiescent follicle. Just to look for the regulation, we are I want to make the difference between K and omega. In fact, omega represents all the growing follicle that can have that can regulate other follicle, and K1 it's the follicle that can be regulated by this follicle. Okay. Okay. So now. I present you this description, a model with discrete description of the size, and we prefer a continuous description of the size, in part because the actual data and the future data, it's okay with the fact that we can take a continuous variable for the size. Okay, so now it was the same type of model, just we are the variable of interest is rho, t is for the time, and x it was the size variable, and it was continuous in 1, 0. So the regulation it's we just change the sum by integral, nothing. Okay, and then we obtain. So in the continuous case, we have a PDE system. In fact, it's a nonlinear transport equation. So the dynamics on the quiescent follicle, it's clear. And the dynamic on the growing follicle, we see the epsilon just before the derivative in time of the density of the quiescent of the growing follicle, and then we see that the transport term of our equation is due to the dynamic of the growth of the follicle. Okay, something it add. It was a condition, a boundary condition, and in fact we just chose a continuous flux in the boundary. Okay, so now we want. We want to understand the limit behavior of the solution of this model when epsilon tends to 0. Okay? So first of all, we propose an appropriate candidate. We think it was an appropriate candidate. So it was very easy. We just take epsilon equal to 0 to obtain this equation. We are two things very important in this dynamics to understand. First of all, it was the dependence of the time of the growing follicle. Okay. In fact, you see that in this equation there is no real dependence on the time. The dependence comes just to the boundary condition, because in fact you have a quiescent follicle depend on the time, but not really the growing follicles, density of the growing follicle. In fact, the density of the growing follicle are at a quasi-steady state all the time. Okay, so it's really the idea that the dynamic of the quiescent follicle is very slow. So the growing follicle have time to be in a quasi-steady state all the time. It's this idea. Okay. So now we just look for a convergence, a theorem convergence for easy case, the linear case. So in the linear case, you have no regulation. So the tau are the rate, sorry, are constant. Okay, and so the quiescent follicle have a easy expression. Okay, and it's the same for the limit case and for the case in lambda. Okay. So then we use a method of characteristic method to solve the equation for the model of epsilon. Okay, and we see that if you are far enough to the initial condition, okay, you have this expression for the density of the growing follicle, and if you are too close to the initial condition, you have another expression there that depends of the initial condition choice. Okay, I just want you notice that the far enough to the initial condition depends here of the epsilon. Okay. And that it was interesting. So we can, you can solve the model, the limit model, and you obtain this expression. And in fact, if you compare the two expression, you can see that if you take care of the initial condition, okay, you have a uniform convergence of the solution to the model in epsilon to the solution of the model of the limit model. Okay. Now, because you are nice, I show you an illustration of this. Wow. So this it's an illustration of the linear case, okay? And then I let Ketum explain show the illustration and the model in the general case. But just to finish with a nice curve, in fact, there it was the density of the different growing follicle, okay? At the axis, you have x, the size. Okay, so you see that for a different value of epsilon, okay, you tend to the curve. There it was the reduced model, the limit model. Okay, and you see that you tend to the limit model. In fact, because of the memory of the computer, we just go to, so the first curve is for epsilon equal to 0.09, and the last it was 0.01. But in fact, we want epsilon at the value is 10 to -4, so we have marge. Okay, so now Ketum go to present you the general case.
Well, before introducing the general case, let's go back to the PDA equation that was introduced on the slide number 8. Well, it's about the resolution of this PDE. So we proceed by discretization in space and time, and we propose here two different schemes to solve this PDE. So the first scheme consists on an explicit discretization in time-space. Well, it's a simple discretization of the PDE problem that gives us this expression for the rho. Okay, just to remark that that scheme involves a stability condition that includes both lambda and mu. And we can proceed differently to solve this kind of PDE by making a change of variables. And here we consider the new variable rho tilde, that this multiplied by this exponentially multiplied by the expression of rho. And then from the PDE presented in the slide 8, we go back to a PDE without any right-hand side that is simpler to solve numerically. And then we obtain this scheme for this new resolution. Well, we can see from this expression that the stability condition for this scheme doesn't involve mu anymore, but only lambda. Well, this is less restricting for us. That's why for the resolution of PDE, the figure we'll present later are with this scheme, because the stability condition is less constraining in this case, since it involves only the maturity rate. So here are some figures illustrating conversions of the PDE equation with the epsilon to the reduced model that is the PDE equation with epsilon equal to 0. So here we have the convergence of the boundary condition rho on 0. So here we can see that the curves are tending to the curve of the reduced model in the point 0, and due to some numerical and computational our very modest computational resources, we couldn't get to epsilon smaller than 10 minus 2. But in general, in biology, for this kind of problems, epsilon is in the order of 10 minus 5. We couldn't reach there, but we can see that from these figures, when epsilon is smaller, we have the curve that is tending to the solution of the reduced model. Here for the boundary condition on the point 0, and here for the growing follicles, we can see also that the curves are tending to the solution of the reduced model once epsilon is smaller. Here we go to epsilon equal to 5, 10 minus 2. So here we have some, we go back to the figure that we presented on the first slide. So we have those biological data that we introduced on the previous slides. And we could, thanks to our model, reproduce at least the same distribution on the follicles growth for the boundary condition and also for the growing follicles. Here we didn't make any data fitting or anything, but just with an appropriate choice of the maturity and mortality rates, we could reproduce this qualitative curve that is quite similar to the curve in biological data. We have this profile with two bumps given here in different times, and we can see that when we get to some age that are important, so we have the diminution of the number and density of population till we get to the exhaustion of the pool. And here we have the curve on the point 0 for the quiescent follicle that is reproducing the same behavior as the curve given here in biological data. So for the conclusions, what we presented here, we introduced a design and simulation of a slow-fast system based on a PDE model. We proved the convergence of this. We proved we proved the convergence of this model to a quasi-steady-state reduced model. So it was the model presented by Céline, where there is no epsilon involved. So this is a quasi-steady state that describe at least the dynamic of the process, and that is a simpler model to manipulate and to solve numerically. We get to reproduce the qualitative behavior that is quite consistent with available data. What we are working on, so the perspective, we have presented a proof of convergence in the linear case, and we can say also that we have a numerical proof of convergence given by those plots showing that the solution for the PDE with epsilon tends and converges to the solution of the reduced model. So we can say that this is a numerical proof, but we still have to prove the convergence when epsilon tends to 0 in the general case. We can do also some quantitative fit-in by making parameter estimation based on biological data. We are working on this part, and I think we will work on the reduced model to do this since it's simpler to manipulate and to resolve numerically. And also, since this kind of population is progressing in a non-deterministic way, maybe a stochastic model can be more appropriate to describe and to follow this population, especially for the population with the small sizes, because the fluctuation in the number of population influence the whole pool when the size is not very important. And thank you for your attention. And if you have questions. This one? Yes. This one?
In fact, we use the simple model with linear case. In the linear case, we have expression for the solution. So we are interesting to have these two curves. Okay? So we find the good parameters to have this one. And then we see that if you had regulation, so non-linearity, in fact, we obtain the blue curve and not the green. And they change, in fact, not completely this one, but more the quiescent, the dynamic of the quiescent follicle, of the density. And we can obtain, thanks to this non-linearity, this type of curve, and we see that the regulation permits to the quiescent pool to be conserved a little bit more. You understand? Okay. Because in fact there is no creation of new follicle, so it's the dynamics just to the first compartment, the quiescent follicle, and when they go to zero, they are equal to zero, it's finished. So in fact there is a very important role of the regulation, because thanks to that you don't go to zero really quickly.
Well, here for the two different schemes, we can see that the stability condition depends on epsilon. So, okay, we should have 1 minus delta t over epsilon delta x lambda minus, okay, we have to verify some condition that is dependent on epsilon. And this condition is verified on the delta t. So when epsilon gets very small, the delta t gets very huge, and then we are running many simulations. Okay, we have to go on many time steps to reach the good solution of the problem. So this is, in terms of computational resources, it's quite restricting for us to go over a very huge number of time steps from one side. And in order also to there is also a restriction about the size of the vector of solution. If it's huge, it takes very, okay, at a certain point we had some memory problem due to this stability condition. But the second scheme is less constraining because it involves only lambda, but it's still dependent on epsilon. So we couldn't get on epsilon very small because it's involving a very huge number of time steps, and we can see that the code is running for a long time. It was not feasible with our resources and with the time we have in CEMRACS, so maybe we can... Yes. Figures, so we... yeah. To do that with the implicit model. Yeah. But we have a code with implicit scheme that there is no stability condition. So maybe we will use it for the next simulation or for the report, maybe. Thank you. Okay, well, this discretization corresponds to the discretization of this PDE given here. So in order to make the discretization, as you know, you put the t in the interval of time tn plus 1, and you put also the space in the interval x I moins 1, xi plus 1, plus 1. And here in this term, when you make the integration of this term here, that involves mu on over the interval xi plus 1, you have to okay. The point was to fix the mu, and then we chose to take the mu value on the medium of the interval, that is mu I plus 1, 2. That's the value of mu on the point between xi and xi plus 1, that is the point, the fictive point xi plus 1, I plus half. Okay? Well, it was a choice. We could choose the value of mu on the beginning of the interval. We could choose mu I, but it's more, let's say, common to take the value of mu on the medium of the interval for the integral. That's what we did. So even the plots of the solution are given on a middle point, that is on the middle of the nodes.
If there is no more question, I just want to say thank you to our three supervisors, because it was a very, very good experiment, and we are very happy to work with you. So thank you very much to Marie Postel, Romain Evinec, et Frédéric Clément.
Okay, I would like also to thank our supervisor. I've said that yesterday. So thank you so much for working with us, and we could discover new things. I'm usually not working on this kind of problematics, but it was quite interesting to discover what can be done in biomathematics. And I would like also to thank the organizing committee for their hostility and okay. Hospitality, sorry. For their okay. So for their hospitality, and thank you very much.
So we start with the biological context. So we are talking about cell segregation and bordering sharpening into species system. So when we talk about cell segregation, we are talking about the organization of biological tissue. So it means in particular in morphogenesis and tissue growth. So we notice that the cells of the same type can regroup into regions, and this means that the cells of the same type can create patterns, so they have a sort of spatial organization. And our starting point is the paper that we see here. That is a work published by a researcher in the Francis Crick Institute in London. These researchers perform a lot of experiments to understand the principal mechanism that influence the cell segregation, that in particular the cells adhesion. That is means that the cells attach each other, tends to attach each other, and repulsion, that is quite clear because that means that the cells get away from each other. So when we talk about inter or heterotypic species repulsion, we are talking about the repulsion between cells of two different population. For example, population of type A and population of type B. And when we talk about intra or homotypic species repulsion, we are talking about the repulsion between cells of the same population, so of the same family. So in this paper, in this work, that also our working hypothesis to modelize this problem, is that repulsion between cells have more influence than, for example, adhesion cell. So the goal is to understand the mechanism of this morphogenesis and this cell segregation, that is important not just to just only for tissue growth, but also for example, the development of various invasive diseases such as tumors and after metastasis. Okay, so. Here, it's not simple to modelize this biological problem. So there are a lot of models that have been proposed and also different approach. For example, the microscopic approach and microscopic approach. So we, for example, for the macroscopic model, we have a continuous approach, and this is quite simple because we have a lot of tools and theoretical framework to analyze and study the model. But in this case, we lose a lot of information at individual level. So, for example, we don't know how many clusters we have, the size of the cluster, and the size of population. And with regard to microscopic model, when we have a lot of cells of particles, it's quite natural to consider our system as complex network, complex system. Sorry. So we can the agent-based model is simple and more flexible for this reason, and also very precise because we have a direct link with the experimental data. But also in this case, we have an inconvenience because this is very computationally expensive and also theoretical harder to study, to analyze. So we start with the microscopic framework that is a model introduced by Diane and Pierre de Gonde and their collaborator. And in this system, we have two equations that describe the behavior of two families of population. So we refer to these as population A and population B. So we consider a set of we consider a set of NA particle or cells and A and B particle of cells. And these two equations describe the evolution in type of each particle. So here we have a two-dimensional Brownian motion with intensity dA and dB that are positive constant. So that is mean that the position of particle change in a random way. And here we have a term of interaction between particles that can be the particle of the same family or the other family. So we can imagine that these particles are able to create links between each other. So each links generate a potential. So I explain now in what I mean with this potential. So here we have the total energy of the S-type particle. So in this case, particle A and particle B. And that is contain the potential. So we are some overall pairwise links potential that act on our family of particles. So in this case, we are imagining that our particles can interact with their neighbors that are located at distance r from their center. So, and this particle acts as a spring. So a spring that is have a equilibrium length also r. So this is the shape of the potential. And so as you can see, we have two parameters, nu c and nu d. That is means that we suppose that our creation and the deletion of links between particles are modeled by a Poisson process frequencies, a Poisson process. So we take into account the Poisson process frequencies. And also the interaction, so repulsion, that we are interested to study between cells. So it's not very simple to follow this particle because the links are not permanent, because we have the possibility to creation and so suppress the links, and this process is very fast. So the principal goal of this project is to add also a logistic term. So that is mean that we suppose that our cells can die and can divide, so can have a daughter, and so the number of particles in A and AB are not so fixed anymore, because change at each time step. So we suppose that the cells of type S, so our population A or B, can divide into cells with probability beta S and can die with probability of delta S. And this process of birth and death depends on the local density of individual of our cells. That is mean that if you consider one cells and these cells has neighbors, that is means that the more you have neighbors, the less is probably that you have a child. So the birth occurs at distance r, a small r, and we see the equation of logistic growth term for the microscopic model, and the parameters are n0, that is the numbers of the cells at distance r0 of our cells that we are considering, and n star is the maximum number of the cell in your radius, so in your zone. And theta is a constant coefficient that assures the randomness at population n star. So before pass to the macroscopic model, we have to go step by step. So it's possible to link the microscopic model and the macroscopic model. And for this, I want to give the talk to Kevin.
Thank you, Marta. So I'll present the convergence of the model from the microscopic scale to the macroscopic scale. So first of all, there has been a proof of the convergence of this microscopic model to a macroscopic model. This proof has been done by Barré, Carillo, Degon, and Diane Perrichard, and Satorska in a paper which is not yet published, in the case without logistic growth. And a second proof, which is not linked to our model, has been done in a case, in a simple case of Brownian bugs, which are animated by a birth and death process. And our goal in this part of the presentation is to merge the two methods in order to perform the convergence of the model. So usually the derivation of a macroscopic model from a microscopic model is split into two steps. So we have the first of all the limit of a large number of individuals and large numbers of links between the particles. And at the second step, we have the limit of large scale and fast network remodeling. But first, in the limit of large number of individuals, we make the assumption that the ratio between the number of links and the number of individuals at the limit is constant. So what is more challenging in our case is that the size of the cell population evolves in time. So it's now more difficult to perform a limit in large scale, in large population. So we need to introduce a new space of probability, which is the space of all the possible states of the particle system. And so we denote this state by a vector Sk. Sk is the vector of containing all the position of the particles. And at a time t, we suppose that we have a k particle in the system. So the probability of having k particle in the system at a time t and in the volume d is k is defined as like that. And with some computation, we can derive the density or the concentration of the cell in the domain by this formula. And after derivation of the formula of the density of the distribution of the particles, we can define a master equation for the probability, for the time evolution of the probability in the Fock space. So this is the form of the master equation. So the probability, we say that the probability of having k cells at a time t plus 2 is equal to the transition probability to be the probability to go from a state sk prime to a state xk at time t plus 2, and times the probability to be at a state sk prime at a time t. So to this we add also these terms which represent the growth process. The first term here is a term that describes the gain in probability at the time 2 due to the birth of a cell at the time where we have just k minus 1 particles in the system, and this here BP is called the birth operator. It is described here. I will explain it after. So here we suppose that we are our tau here is small enough such that we just have one birth process or death process in the time. So here this term describe the fact that we lose probability in the configuration with k cells due to the birth of one cell or due to death process. And this last term describe the fact that we gain in probability in the configuration with k cells, when in the configuration with k plus 1 cells there is a death So this transition probability, we should expand this transition probability and describe it using a formula called Cramer-Moyal formula to expand this transition probability. Now, this birth operator is used to describe the process of birth in a configuration with k-1 cells to a configuration with k cells. So this delta function here is a Dirac, which means that a parent cell should give birth at its position. So here, so after taking the limit when tau goes to 0, we should get an equation in the derivative in time of the probability, and from this equation we use the formula for the density and the expression of the transition probability. We didn't put it here because it's a very big formula. So you put the expression in the equation, and so you integrate, and by summing the equation, you should get a kinetic equation. And this kinetic equation, to obtain from this kinetic equation the macroscopic model, we perform the rescaling of the equation. So we rescale in space and time with an epsilon, and we take the limit when epsilon goes to 0, which allow us to derive the macroscopic model. So after this derivation is just a sketch of the proof. So there's no more details about this. But after the derivation process, we should get logistic model at the macroscopic scale. So this logistic model is expressed as like this. This equation is known to be Makin-Vlasov equation, but plus logistic term here. And this term is the term of interaction, which expresses the links between particles, the potential between particles, and this equation is the equation on the evolution, on the time evolution of the density of the species A, and the second equation is the time evolution of the density of the species B. And here I will more explain the logistic term. So in the logistic term, we have here a term which expresses the growth of the population. So it is a saturated growth. And F star is the current capacity of the environments. And new AB is the growth rate of the species A. And new BB is the growth rate of the species B. So for the next step, in which we will analyze the model and will perform stability analysis on the model, I will pass the talk to Remy.
Okay, so as Kevin said, now we are very happy because we have the macroscopic model, and with the macroscopic model we can do a lot of theoretical work. And so if I remind you, the goal of this project is to have a model, access to a model that allows you to see some cell segregation and aggregation. So you want to know, okay, we have the model, but this model can give you really aggregation. And to know that, we can perform some stability analysis. Why? Because you have a steady state for this model, which is an homogeneous one, where the density are the same everywhere. And you want to know if this state is stable or not. Because if the homogeneous steady state is stable, then you can't hope to have some aggregation. Whereas if the steady state, the homogeneous one, is unstable, then you can hope to see really aggregation in your model in a long time. So we perform some stability analysis. What you have to remind is that the homogeneous steady states are defined with this equation. which means that everywhere you reach the carrying capacity. And to do the analysis, we do some linearization around the steady states, and after that we do some Fourier transform. This is the equation that you get after the Fourier transform. You see that you just have here a big matrix, and you want to know, okay, to know the stability, we just want to know the sign of the eigenvalues of this matrix. And we have access to an easy criteria, which gives you which tell you that the homogeneous steady state is unstable. Okay, but in this project, we want mainly to focus on the ratio between homotopic and heterotopic repulsion. And so we set a parameter here, which is a parameter S. You just want to remind that the parameter S gives you this ratio. So if you have, you take a big S, then that means that heterotopic repulsion is stronger than homotopic repulsion. If you take a low S, it's the contrary, and so you have an homotopic repulsion which is stronger. Okay, and so when you do that, you can in fact find a critical value which we set here, which is S star. And if you take a value of S which is greater than this S star, then you can say that the steady state, the homogeneous steady state, is unstable. And if you take S lower than this value, of course, the homogeneous steady state is stable. So that's very logical. You just say that if you want to have aggregation, you have to be very mean with the others and not so mean with your family. And you say, okay, we have this criteria, but in this criteria you have the value of the population at the homogeneous steady states that appears. And when you look at the homogeneous steady state, you say that this equation is satisfied, but to satisfy this equation, there is in fact an infinite number of steady states. So you say, okay, we want to know how this parameter, this critical value, depends on the steady state. And so with this equation, you can say, okay, I can shut down all the FB in this equation to only have an equation on FA. And we find that this equation gives you a rational function. And you say, okay, we want to study this function. Okay. Okay, there is a lot of information in this slide. So here we can see some plots of the value of S star depending on the question of the concentration of the family A at the steady state. And so you can see that in fact this parameter behave like that. That means that this criteria goes to infinity for very low population and very big population. That's a very bad aspect, because you can always say that it's impossible to have all the steady states that are unstable. If you choose a value of S which is like that, you will say, okay, there is the low populations and the big population which are stable. But the good aspect is that if you observe the behavior of the criteria, you observe a very flat shape here, saying that for at least for a reasonable value for the parameters, that means for biological values, that means that you can still hope with a value here to have a great part of the steady state to be unstable. So if you hope to begin near around this concentration for the population A, maybe we can hope to have aggregation if you take S strong enough, because close to this value there is a very flat plateau. Okay, so if you do that, you can also do the same work with the model without logistic. So the study of the stability analysis was already done by Diane Perecha and the collaborators in their first study. So we just remind here the value for the criteria. Of course, it's not the same. And we want to compare the value here and the value here. And what we have found is that there is not a really easy way to compare the both, because all cases are possible, meaning here in the plots, in fact, it's in red you have this one, and in blue you have this one. So you can see that with some parameters, the red one is greater than the blue one, but also you can have the contrary. The blue one is greater than the red one. And you can also have some mixed situation where they are very close, but at a time it's the blue which is greater, but after it's the red that it's greater. Okay, so there is not an easy comparison between the two criteria. And so what we get from that is that depending on the parameters, logistic growth can either support aggregations or repress aggregation. Okay, so the situation is not very simple, and you want to see what happened during the simulation, the numerical simulation. And so we performed some numerical simulations of the microscopic model, because the idea is that if you do some simulation of the microscopic model, you can choose the parameter to see if you find back the results that are given by the stability analysis. And if you really find back the results given by the stability analysis of the macroscopic model, that means that the microscopic one is really a good approximation, is really linked to the macroscopic model. So we place ourselves in this case. That means that it's a case where the repulsion of A on B is greater than the repulsion of B on A. So the population of A push the population B stronger than it push itself. Okay, and with this parameter, we want to study the impact of the logistic growth on the different behavior at long time. And so, in the first case here, we choose to have the population A, B, which divide faster, and in the case 5, it's the contrary. It's the population A that divide faster. Okay. Okay. So this is the first case. When you say that the population A repulses the population B stronger, but it divides slower. So there is a balance between the two actions, and so you hope no aggregation, because, okay, the A are stronger, but more or less the B are, there are more B. Okay. So in fact, in the analysis says that with this parameter you can see aggregation without logistic, and it's really the case. And the analysis says you cannot have aggregation with logistic growth, but in fact we observe some aggregation in this case. A thing that I've not said is that in all simulation we begin with 250 cells of type A and of type B. Okay, in the second case, you have it's still the same. You are supposed to observe no aggregation with the logistic and aggregation without the logistic, but in this case, the population A and B divide in the same way. And so again, we observe some aggregation with the logistic growth, which is not predicted by the macroscopic analysis. In the third case, we check that if we hope to see no aggregation for neither with logistic or with logistic, it happens. So there is no aggregation here for the without logistic. And with logistic, it's not so clear that there is no aggregation. We can still see some big shapes that appears. And if we force the model to give aggregation with and without logistic, we observe aggregation with logistic and also without logistic, but there is the shapes are very different. We have very big cluster with logistic growth and small one with without. And then you say that the population of A divide faster, so they repulse more and they divide faster, so you hope to have a very big clusters for the population A, and that's really the case. There is a big clusters for the population A, and without logistic you hope no aggregation, and in fact there is no aggregation that you can see here. Okay, so for the conclusion I give the talk to Kevin.
Thank you, Rémi, for giving me the talk. So for a conclusion, we present a two-species model for cell segregation, and we take into account in our model microscopic approach and macroscopic approach. We focus on the influence of heterotypic and homotypic repulsion, and we add a logistic term to our model, and we study the logistic growth effect on the stability of the steady state. We perform numerical simulations of the individual agent-based model to confirm the result provided by the stability analysis. And after, in perspective work in progress, we have already done some numerical simulation of the macroscopic model, but not yet. We are not understanding what we obtain. So, so in perspective, we are hoping to improve this numerical simulation and to understand what we get as a result. And our aim is to rigorously derive the macroscopic model, and also to understand the differences that we notice between the macroscopic and the microscopic simulation results and the stability analysis. As Rémi said, in the case where we are not supposed to have aggregation with the logistic growth, in the macroscopic model we have some aggregation, and we don't really understand the cause of this aggregation, and our aim is to understand it very well. So thank you for your attention, and
We can't, but they can't, because Diane, in their papers, they do some criteria that allow them to quantify the number of clusters and the size of the, more importantly, the size of the clusters. But we didn't have the time to do that. It's possible, at least for the macroscopic model, it's done, but not for the. We didn't have to look at that in this case. Yes. Yes, we have some difficulties because the first one, which is the obvious one, is that you want to see the behavior of the model at long time, so you have to. The computation are very expensive, and we have problems of cost of the computation. So you want to do some. You want to be a little clever when you do the numerical schemes, and it's hard. It's long to implement. And the other part is that you want to do a numerical scheme that ensure you that you preserve positive quantities, and it's a little bit tricky because you have to be cautious with the term of interaction, and so we have some difficulties with that because we read a paper that Diane gave us to do that, but it involved a CFL condition to ensure the positivity, and so you have to take an adaptative time step, which can be very low when you perform long simulations, so it's quite hard to do. And the simulation for the moment are not so satisfying because we are not so sure to preserve positivity of the solution. For the parameters that we choose, the process is not is a continuous process. But I know that in their papers they do some analysis. So when we choose to change the parameters, can you have a phase transition for the segregation to appears? But we didn't look at that at all. And in the simulations that we perform, there is no the aggregation process is continuous, and so from the beginning to the end, you see aggregations that are growing. Yes. It's not at all taken into account in the model. We really don't know so much because, in fact, it's hard to say that. But in this model, you don't say that if a cell is behind another, we can create a link. That's not said at all. But in fact, in this model, the parameter R is taken. The long length to create a link is taken from about the radius of a cell. So if a cell can interact, so they are really touching each other.
Okay, it's just a little announcement for the next CEMRACS. That okay, I didn't put everything because I couldn't, but just to give an idea of the kind of topics that we want to address. So basically, you have like oceans and atmospheres and the coupling between the two of them. Okay, you have the like also river flows are totally in the topic. So transport of pollutant, for example, or you see that we are also talking about ecological aspects. So we encourage like cross research areas domain projects. And you can also think of porous media, like Darcy equations, for example. They are totally in the topic. And I put also granular flows, so avalanches, dune formations, and you can also couple granular flows with river flows if you want. Well, also basically this kind of things, okay? And I didn't put everything. You can also do data assimilations on this kind of topics or stuff like this, okay? And so the first week courses are already set. And if you have some questions, like if you want to participate, you have some questions, or if you want to submit a project for the next CEMRACS, and you have also some technical questions, please send us an email to the email address that I put there, or you can also send an email directly to one of the organizers. Okay, it's okay. And so yeah, feel free to send an email to have some informations. Okay, or I'm here also if you want today or tomorrow. Okay, thank you.
Yeah, so, okay, thanks. Well, we'll start with thanking all of you to come here and listen to us. So our project is about horizontal gene transfer, and just a few words about what is it. Formally speaking, it's exchange of genetic material which happens not through vertical transmission, that is from parents to offspring, but just due to exchange of material between living organisms. Here we have just a scheme how it looks like approximately in a biological context. So we have here just two bacteria, and one of them is carrying some plasmid, and after exchange, this plasmid also is transmitted to the second bacteria, so that they become identical. That's how unilateral horizontal gene transfer looks like. This phenomena is very important in modeling, for example, evolution of organisms, transmission of pathogens, resistance to antibiotics in bacteria, and so on. And since we are not biologists here, for us it looks more or less like this. So, well, here what we have, in fact, on this plot we show evolution of big population of bacteria. So every point on the plot is just some individual. And when we see strains like this, then in fact what's going on in the strains is that some, let's say, cool individuals who can transmit their information to the other, they just drive the whole population to some other traits, which is kind of just very common generic name for genetic information encoded in these individuals. So the whole population becomes different, not very fit. And then when we look, for example, when we look on this point, then we see that the whole population goes kind of to extinction, because we don't have any movement forward. And then starting from this point, again, all the individuals, they kind of regrow and repopulate again environment, and again, due to this gene transfer, they become less fit, why we will explain it later, and again die and regrow. So this phenomena is very interesting to model, and that's exactly what we're going to do. Before we move to some specific models, we will just talk a bit about how the whole demography is regulated. Again, trait, which is just genetic information for us, it's some numerical parameter. Also, we take into account carrying capacity of the system. By that we can mean, for example, amount of resources in some environment. And then when it comes to individual characteristics, then we have birth, death, and horizontal rate, horizontal transfer rate. Here, well, birth rate is more or less obvious. But about death term, then this thing in fact comes from competition rate. So we just know that all our individuals, they compete with each other all the time because of resources and things like that. And horizontal gene transfer for us is just some function, heaviside or stuff like that. So it will be discussed further. And the aim of our whole project is, in fact, to describe this population on different scales with the help of different models, and just understand how those models link to each other, and well, if all of them are able to catch this behavior which we observe, and in let's say further work, if to provide some theoretical explanation to what we see just numerically. So first model in this on this scheme is stochastic model, which is the most straightforward one. It's based just on individuals, so we just model exactly all those individuals. They can die, they can give birth, and so on. And it makes sense to consider this model when we have, well, relatively small size of the whole population, because when it's too big, then it makes no more sense to think about each member separately, but to consider the whole population. And in that case, this whole population is described by a deterministic model, so we just speak about densities. And if you want to move even further, like when we want to consider very big time scale and very small like mutation size, then we move to Hamilton-Jacobi PDE, which will be also explained a bit further. So then our objective, in fact, to study first all those models numerically, and as intermediate step, we will have also to develop a stable enough scheme to deal with vanishing values, and that's what Alan Ivar did for us. And then also analyze the setting by which we can observe either extinction or cycling behavior or just some oscillations from theoretical point of view. And I will talk about stochastic one, which is described by horribly looking equation of on 3 lines. But well, even if some of you are not familiar with like jump processes or something like that, it's not so hard to understand what's going on, like I hope. Just what is written here is that if some individual gives birth, then in fact we add a new individual with a bit different, like mutated trait to the whole population. So we consider the case when we have mutation at every moment, at every birth, and this well, okay, and this mutation is regulated by this like mutation kernel. In our case, it will be just Gaussian kernel, so we have like mean value is initial trait, and then just some small variance. It's like trait of an offspring. Then this term is regulated by this. We have already seen it. For us, like this thing will be just like some like square function, like dx square. And horizontal transfer, for us, it will be just Heaviside. So when horizontal transfer happens, what we have to do is to remove individual from remove one trait in our system and then replace it by another trait, which was given by a donor. And if we want to model it, then let's just also, well, it doesn't look very nice and easy, but in fact it's not as difficult. What we have to do, we just compute arrival times of each event according to exponential law, basing on the rates which we know from, well, which we just said. And then, like, if each arrival time is smaller than our time step, then we either remove one individual or like if you know that it is going to die on this time step, or add a newborn to the whole population, or replace one trait by another. In this setting, we assume that all 3 events can probably happen with the same individual. So like we don't take into account that, like, I don't know if it's that, like, it cannot give birth anymore, but in fact if this time step is small enough, then like we don't care anymore. It's still quite accurate. And then depending on the parameters that we introduce, we can observe 3 types of behavior. So it's either stabilization. It happens when like our whole population just stabilize around some optimal mean trait and like stays like this for the lifetime. So nothing really happens in there. They don't go to extinction. They like don't compete much, and like that's the most maybe boring case. So in second case we have cycles. Maybe on this picture on big screen it looks a bit like some diffusion, but in fact we have really some cycles which are like quite extended in space. And also if we see here, if you look at the plot with mean population size or mean trait, then we also see that like it's cyclic. So maybe it's it looks a bit better. And that's maybe the most interesting case, because when we speak about jump processes, it's not completely clear how to describe it from theoretical point of view. And that's why we will focus on this case going to deterministic models. And finally, if our transfer term is too high, then the whole population just is driven to extinction before it can repopulate the environment. And also, it was mentioned that in stochastic case, again, we know that when this value of transfer is very close to some critical, which again we can only estimate empirically, then we can observe in fact both types of behavior. Sometimes our population manages to like adapt and show cycles, and sometimes it goes to extinction. So yeah, because we have just some random events. Yeah, so how, if you are interested, then like you may look at even more pictures on our GitHub repository, and then I will just give a word to Suzy, who will talk about PDE model.
Well, thank you, Anya. In fact, it's really usual to derive a macroscopic model from when we have a large population in the limit of, in this case, in the limit of k, which is our population scale, to go to infinity. For our cases, we have some integral differential equation, which is given with more or less one depth term, one burst term, and one transfer term, as in the previous stochastic model. Here we have that D is this rate, and we have the competition rate C, which is multiplied by the total population size. We have also one integral term that is representing the burst rate, which is also multiplied by some mutation, because Anya is already explained that when we have bursts, we can have some mutation in the new individuals. And for the case of the transfer term, we consider that it's the product of some constant tau positive with a smooth function alpha that we can use Artangent or Heaviside as usual. This kind of model, we can, if you want, you can read more about that in Villiers and its collaborator, and Ferrier and Tran in 2009. Sorry, I didn't say that in this case, the density of population is modelized by F, which is the density at time t. For population, we have a trait X. For our PD simulation, we are first interested in compare if we have the same kind of behavior that for a stochastic model, and if we keep the same kind of parameters. For that, we implemented some explicit error scheme, which for which the integral terms in the model are simulated by left point quadratures. And we recall that what we have is the competition term, which is the total size of the population, which is also computed in an explicit way, and the death rate, the transfer rate, which is in this case the function tau multiplied by the sum of the function tau multiplied by this fraction of total population with density. And the important part in this, the important term in this part, which is the burst rate term, which is a convolution for which we need to make some grid also in Z, which is made in the same way that for the discretization in space and time that we used to make in this kind of explicit scheme. And what it's important to mention here is that for the value of f in the point xi plus zk, we make some linear interpolation if this value is inside of the x grid, and some linear extrapolation in left or right slope if it's outside of the x grid. With this kind of scheme, we can obtain the same with some different kind of species. In the first time, we have for the value of tau equal to 0.2 some cyclical value, but some cyclical behavior, but until the population can stabilize around one mean rate, which is the kind of behavior that we had in the stochastic model. Moreover, for the case that tau is near to the critical value, we say the 0.4, we can see some oscillations that, some damping oscillations that until one moment, starting from one moment, they become some dumping and vanishes around some stabilization, some mean rate, which is also observed in the mean rate plot and the total population plot. And this damping oscillation need to be understood in the way that maybe the scheme is not designed to grasp the exponential small value of the density population when the cycle occurs. And for that, we propose a better scheme for the sequel. And also, we observe that for bigger value of the transfer rate, we have also some extinction phenomenon, but in some moment of the time, it seems like a population can be able to regrowth and to have some vanishing behavior around the stabilizing the mean rate. Here we observe also that we have some period of time for which we have not a population at all, and after it's able to reproduce it again. I let Sam to explain a little about the sequel of the.
Yeah, so even if in the stochastic model we were able to see cycles and beautiful oscillation, as Susie explained, in the PDE model we are not really able to, we see oscillation, but the oscillation are fading, and thus we are not able to numerically grasp the phenomena we want to modelize. And even theoretically, from the PDE model itself, it's very difficult to have explicit formula for to have a precise description. So we have to push further the analysis on the theoretical aspect, and it will give something numerically as well. So we have to understand a little bit what the solution look like. So what is the shape of the solution we are trying to compute? So first of all, there is one important feature in our model, is that there is a competition a competition term, meaning that all the individuals compete with each other for the same resource. And this leads to a saturation phenomena, which mathematically speaking means that the total population, the total number of individuals, if you want, is bounded. It remains bounded. Okay? And saturation phenomena leads to selection of the fittest individual. In the context of competition, you have saturation, and therefore only the fittest individuals, so those who are the more adapted to the environment, can survive because of the competition. And so our density will concentrate, will converge to a Gaussian, to a very narrow Gaussian, centered at some trait x bar, which is the most adapted trait. So this is what we expect from the solution, okay? And the variance of the Gaussian will be of order of the variance of the mutation kernel, okay? Without mutation kernel, we expect F to converge to a direct mass. And so to push further the analysis, we decide to make the mutation kernel very small. So we introduce a rescaling parameter, epsilon, to rescale the mutation kernel. So from a mutation kernel who looks like this, we rescale it by epsilon in order to have something which is more narrow. Okay, so we reduce the size of the mutation rate, of the mutation kernel. And on the counterpart, to see the effect of very small mutation, we have to look for large time. So we also rescale time. So we have two time scale in our model. Then we have the normal time scale, which is the ecological scale, and the evolutionary scale, T over epsilon, where we can see the effect of mutation. And so the partial derivative, mathematically speaking, is transformed to something like this. And so from the PDE model, the rescale PDE model looks like this. Okay, so it's the same thing as before, just with a rescaling. But again, to be able to pass to the limit when epsilon goes to 0, we have to remember that, okay, I recall that we expect F to be of the form of a Gaussian of variance epsilon, which is the size of the mutation. Okay, and so we perform the celebrated Hopf-Kohl transformation, which is we change our variable. We don't compute F anymore, but U, which is kind of exponential singularity, or I don't know how to call this, but as we expect F to be a Gaussian of variance epsilon, it should be convenient to consider this function U. Okay, and the equation that U satisfies is this one. And in fact, we're able, theoretically, at least formally, to pass to the limit to this equation, with this equation, and to see that at the limit, so formally at the limit U will converge to some function, which is in a weak sense solution of a function, of a PDE, which is the Hamilton-Jacobi PDE that we were talking of in the beginning. So this is the main equation I will be talking of. So what is the interest of this equation is that the interest that it describes very precisely what we want to see, and we are able from this equation to derive theoretical results. And so just to give you an idea of what we can do, not going into the details, we have a criterion for extinction, okay, with computing the maximum value of our function U, okay. We can also have information on the location where the population concentrates, so namely the fittest trait, so the most adapted phenotype, okay. And yeah, I just mentioned that in the classical case we have this equation, but this term, which is due to our specific horizontal transfer feature, is non-standard. So it is the main difficulty of our approach, okay? And so from Hamilton-Jacobi you have a lot of results. So you have a formula for the total population. You can have adaptive dynamics, so the equation, the evolution of the location of the fittest trait. Okay, it solves an equation. We can formally compute the evolutionary equilibrium, which should be 0 if we discard the effect of horizontal transfer, but in our case it's not 0. Okay, And we have also threshold for having oscillation or having extinctions. So this framework is very convenient for theoretical analysis, and I will let Suzeli explain to you how it is also convenient, numerically speaking, to grasp the oscillation we want.
Thank you, Sam. In fact, if we want to be able to catch the concentration, the Dirac concentration phenomenon, we need to make epsilon goes to 0. It's translated to refine the grid in X. There is make the space step decrease. With this kind of framework, the PDE scheme that we had before is not consistent anymore, and we need to implement some asymptotic preserving, we call it HP scheme, which is proposed in the end of the last cycle, the last century, pardon, and which permit to avoid the increase of computational cost and ensures also that the scheme approaches well when the small mutations can happen and to derive in the Hamilton-Jacobi equation. One way to understand this AP scheme is that when we have some epsilon bigger than 0, the scheme in the AP scheme is consistent with the PDE problem, and when epsilon goes to 0, normally the solution of the PDE problem converge to the solution of the Hamilton-Jacobi equation. There is the P0, the solution of the P0 problem. And as the scheme is asymptotic preserving, we will be able to keep this the scheme stable around this asymptotic transition, along this asymptotic transition, and to get some scheme in the limit which is also consistent in which some discretization parameter fixed, which is also stable with the limit problem with the Hamilton-Jacobi equation for smaller tau. In fact, what we do is some numerical AP scheme for epsilon really small and not 0, but smaller. And we recall that for u given in the be the logarithm of the density solution, we compute one almost explicit scheme. We recall that P is the total population size. And in this case, we need to compute the total population size in an implicit way in order to keep the consistency of the scheme with the limit problem. So for the rest of the parameters in the scheme, we just keep some explicit scheme, the disk rate, the this term, the burst term, and also the transfer term. They are also computed in an explicit way. We remark that for the computation that have some exponential term, we need to be consistent with the preserving stability of the scheme. And for example, we can notice that if we want to this quantity to converge to a Dirac mass, we cannot compute the integral directly, but we can make some factor about the maximum value of u that we assume that is take it at just 1 point. And like this, the argument of the exponential, there are just non-positive, and we can be able to deal with this scheme when epsilon is smaller. Then with this scheme, we are overall the same kind of population, the same kind of diagrams. We see some stabilization pictures when tau is around 0.1. There is the very small transfer rate. Then the population stabilizes around some mean rate, and we don't see more phenomena that so different from before. But for the case of the cycle, we see that we are really precise in this case. We have not more damping oscillations anymore, and we see that it is a kind of periodical behavior around the total population, which is able to recreate and reborn and have a periodic size in this sense. And also for the mean trade, we have seen some cycles, which is really observed in these pictures. This is also for the value of the parameter, which is near to the critical tau. Moreover, which is really important to catch in this kind of framework is that for the value of the parameter that we can be able to get extinction before, we in fact got extinction for some value of time. We see that there is some periods that the solutions, the size of the population is exactly 0, and after the population is able to regrow, and it can be explained in the way that we are able with this scheme, with this model, to catch the phenomenon of the evolutionary rescue that I would let Sam to explain a little about formally what is we can obtain.
Okay. Yeah, so just to sum up and conclude a little bit, so what we've done is just starting from a stochastic model where we are able to see oscillation and perform 2 limiting procedures. So first going to PDE for large population, and then going to Hamilton-Jacobi for rescaling parameters that goes to 0. And so after 2 limiting procedures, we've shown that we are still able to grasp the feature we want, namely the oscillations, the oscillation phenomena. And this is important because on the Hamilton-Jacobi level, it's easier to perform numerical tests and theoretical analysis. And it's, I think, in my opinion, an illustration of the fact that sometimes you have to understand theory to perform numerics and vice versa. You have to do both at the same time. And what is maybe new what is new maybe in this approach is that Hamilton-Jacobi has been used for almost 15 years to describe the concentration phenomena, so where the dominant trait is located. But here, the numerics convey the fact that it's also able to give a precise description of a small population, because oscillation are rely on a good description of small population. And so these numerics we showed you open paths to, I don't know, to a lot of things. So yeah, just to sum up, so okay, this is the these are plots on the stochastic level, so of the population at 3 chosen time. Okay, and this is plot which on the Hamilton-Jacobi level, which describe, okay, which correspond one to each other. So the positive result is that Hamilton-Jacobi should be able to give a precise description of the stochastic model and of the oscillation phenomena. And the open question are, of course, rigorous proof on the Hamilton-Jacobi level, formula for the period of oscillation, which is intricate because it involves the time. We have to compute the time for the rescue of small population. Okay, and one other aspect that we wanted to show you, because it's funny, kind of. It's if we compute lineages. So we wanted to see. So okay, this is a plot for stochastic model. So each point is an individual over time. Okay, and we wanted to see at final time what were the ancestors of each individuals. So the lines that are drawn here are lineages. So all the ancestors of each individual. And so this picture is, so the first thing we note that there is only one common ancestor, meaning that the whole population is this children of just one individual from the beginning. And it also there is the idea here that the only people the only individuals that were able to survive were the very few individuals that remained preserved from the horizontal transfer. So there are very few individuals that we are not even able to see on the stochastic picture, but they are there and they are responsible for the population. And we think Hamilton-Jacobi could help to understand this, because we hope that this curve should correspond to the characteristic of Hamilton-Jacobi equation. And okay, references. And we thank you, all of you, and the organizer for hosting us this summer, and you for your attention. Thank you. Anyone?
It was not an easy voice for that.
Yeah, so well, just a small reminder that again, Alan helped us a lot with that scheme, so yeah, that's why like it's not that nice that we kind of didn't stop on that path precisely. So yeah, in fact, the hardest thing to build is a mutation kernel, because and also to compute the value of rho, because while rho is defined explicitly precisely for PDE model, for this model, like we cannot just leave it like this and say that okay, well, that's integral over the whole like population size. So because if we try to compute it from exponent, then it will always give us some degenerate values, right? So we like the method which we used, in fact, allows us to compute this rho from just a boundary condition on the of the PDE equation. So we just know that okay, well, our solutions are bounded, and from that we kind of derive the formula for rho. And in fact, also it's an interesting question, like what exactly does it mean that when we have like very small rho and maybe even zero, because like in PDE approach it would like it could be explained just by total extinction of the population, but in fact here it's not the case anymore, because like even when it's zero, like numerically, it still doesn't mean that our population cannot regrow. Yeah, so in fact that's an open question, like what exactly our rho means in the, well, in this setting. But I'm not sure that I can now recall all the details like by, yeah, so I don't know, maybe someone else. But drift.
Yeah, so in absence of, so the parameter we choose are such that in absence of transfer rate, if we discard transfer rate, the optimal trait is 0. Okay, but now you have a transfer rate that pushes the population upward. So for what we see here is that for a small value, so okay, let's focus on this picture. So in fact, you see that the mean trait is not 0, it's pushed upwards. And this is because of transfer rate. Because population, I mean, people with a higher trait infect the other. So in fact, the optimal trait is this one. So you always concentrate on the optimal trait, but now the optimal trait you have to take into account the transfer rate. So it's shifted. And yeah, and what happened here is that the optimal trait jumps. In fact, the optimal trait is pushed towards high values, and reaching to a certain point, this one becomes more fit. So the population jumps to this one. Well, I think if everything is symmetric, we should have a symmetric behavior, but it should spread a little bit the population. It will play the role of a mutation kernel. Okay, yeah.
Yes, thank you for the introduction and to give us the opportunity to work on this topic. We will talk about the adipocytes, and we want here to model their dynamics. So first, what's an adipocyte? Let's say that that's a bank of fat in the body. These are the cells that store the lipid droplets that your organism does not use for the daily consumption. The talk will be divided in 5 parts. First of all, I will present the motivation for studying this dynamic. Here you can see the evolution of the prevalence of overweight in different countries in the past and some projections for the coming years, actually in only 2 years. As you can see, many rich countries are subject to a growing of this the proportion of overweighted and obese people. Well, that would not be a problem if obesity and overweight was not related to more disease and death. Here are the cause of death in the United States. In red, you see the ones that are related to obesity. You can see that is not neglectable at all. So that's why we want to study the dynamic of adipocytes. First, we will consider a 0D model. 0D meaning that there is no space. First of all, here are the biological considerations. On peut lancer. Okay, here is the video that show how the lipid droplets are stored in the body. Mais en grand écran. You can see in yellow there the lipid droplets then enter inside the adipocytes and then make them grow. Here the spatial consideration is obvious, but we will first close this window and then set aside this space consideration at the moment. Okay. Here is a representation of here are the mesenchymal cells, which are cells that can differentiate into different kind of cells. Here, this branch is the one that interests us, with the mesenchymal cells differentiating into pre-adipocytes through adipogenesis, and then differentiation into adipocyte. So we will have a three compartment model. Here are the mesenchymal cells, which can proliferate and die, and differentiate into pre-adipocytes, which can also proliferate and die at different rates. These cells are roughly spheroidal of a certain radius, and when the pre-adipocyte differentiate, like a radius constant in these two compartments, and when pre-adipocyte differentiate into adipocyte, they appear at a certain radius, and then will grow as the lipid droplets come inside. So that's why we will structure the adipocyte population by radius, and consider that there will be no more proliferation for adipocyte, but there will be a dying rate, and of course a grow at speed v of tr. And then the last interaction is a retrocontrol through the mean radius of the adipocyte over the differentiation rate of mesenchymal and pre-adipocyte cells. That leads to a mathematical model, which is this one. Here we have two ODEs for the mesenchymal cell and pre-adipocyte cells, not involving the radius. As remember, I told you that the radius was roughly constant for these populations. In red, we have the death term, green proliferation, red there too. In blue, the differentiation and retrocontrol term. Here we have a flux, which is, well, the growth of the adipocytes. Here on R star is the minimal radius I told you about. At R star, the new adipocytes are the pre-adipocyte that differentiated. That's pretty logical, I guess. Of course, we have initial conditions. Furthermore, well, here is an expression of the mean radius, what I meant by mean radius. Okay, so here's a coupled system of ODEs and a PDE. To finish with, I have two last functions to describe. The velocity here. This velocity depends on the K of t, which is the excess of available lipid droplets at mt, and S of t, which is the total surface of all adipocytes. Because you can easily understand that the larger is an adipocyte, and the more it will receive droplets. So we have here this fraction, K of t over S of t. And then we have an indicator function here that with a RC here, which is a critical radius, that accounts for the fact that we do not observe experimentally adipocytes of radius larger than RC. That's why we have this indicator function. So that provides a positive, a non-negative velocity. And to finish this part, we have the shape of the retrocontour, where we take a sigmoid, like roughly of this shape, that accounts for a saturation phenomena. This sigmoid has this expression with these parameters to be chosen. And that's all for the modeling. We will have then mathematical analysis presented by Pascal.
Thank you, Hugo. So I now will define more precisely the mathematical framework. Indeed, since we are looking for a solution as, for example, a finite measure, and since V is discontinuous, the product VA is not defined in the sense of distribution. So we need a special definition for a solution. Fortunately, the velocity is one-sided Lipschitz. That means that this inequality holds. And in our case, V is even decreasing, so it is this velocity, this inequality is easy to check. And in this setting, we can define characteristics for the transport equation thanks to the Filipov theory. So in one dimension, Filipov theory is equivalent to say that this system for the characteristics have a unique Lipschitz solution when this line is a rigorous expression of the fact that dx over dt is equal to v. So this expression seems a bit complicated, but in our case it leads to very natural definition of characteristics, and we'll see it on an example. So here we take a simple example where the velocity is discontinuous, but is assumed to be constant in time. It gives this characteristics, this form for the characteristics. For example, if we take a point here, the velocity is 1, so it will move with velocity 1 until it reaches the critical radius, and then it will stop and stop growing. That's why we have a lot of characteristics that merge into this particular line. In our case, when the velocity depends on time, we have almost the same phenomenon, but instead of straight line in this part, we will have a curve, parallel curves. And we even can write explicitly the formula for the characteristics. And it is one of the reasons why this framework is very convenient to have explicit computation. Once we have the characteristics, we can we can define more precisely what we will call a solution in a Pupo and Rask sense. And in your case, roughly speaking, a solution is something that verifies the Duhamel formula here. So this Duhamel formula can be understood if we say that a weak solution, in the case where we have a continuous velocity, verifies this formula. So since the product VA does not appear anymore in the formula, we can generalize it to our setting. And with this framework, we can have some interesting theoretical results, and in particular the existence and uniqueness of a solution to the transport equation based on a fixed point method. And it is not obvious because the velocity depends on the solution and also the flux at the boundary. We can also have some necessary and sufficient condition for the existence of a stationary solution. And we'll see it later. And then we can have asymptotic behavior in some simple case. For example, I represent the case gamma second is equal to 0, and here the evolution rates that correspond to the kind of growth rate in the ODE are negative, so we expect the cells to decay exponentially. And we will have that the repartition of adipocyte at time t will tend to a Dirac mass in RC. And then why this case is interesting, it's because on the simulation that we will present later, we are almost in this case with a very small gamma second. We can also, when there is stationary solution, look at the form of stationary solutions. So the form is first a term of an exponential decay in R, plus a Dirac mass in RC that correspond to the accumulation of adipocyte at the maximal radius, and with two numbers, lambda and v, that depend on parameters in an explicit way. However, if we look at the stability of stationary solutions, then so in this figure, I represent the number of mesenchymal cells, the number of preadipocytes, and the total number of adipocytes, with initial values that are set for the stationary solution. So m is equal to 0 in this particular case, and for p, it is not 0 at the beginning, but the scale is very big here. And we have an explosion of p and a. So it may imply that the stationary solution are not stable, but we need to check. This example is with one particular set of parameters, and we need to check with more parameters. So now we'll present the numerical analysis.
Thank you. So once we have theoretical results, we can do before, but now we have the theoretical results, we move to numerical analysis. In our simulations, we used a simple and the simplest explicit Euler scheme that you could think about. For here it's for the ODE on the mesenchymal cells. Just like here is a discretization of the derivative, and there for the right-hand term, which leads to this expression for the next time step. In the same spirit, we have this expression for the p index k plus 1. And of course, this is conditionally stable, like positive, I mean, because this might be negative depending on the values taken by beta of rk. So one idea we had to change to deal with this problem was to take a semi-implicit scheme. Actually, we did not implement it yet, but we will do this in a near future. So for the semi-implicit Euler scheme, here is the semi-implicit part, and this leads to this expression for m index k plus 1, which ensure positivity. Well, of course, we assume that alpha minus gamma is positive, otherwise the mesenchymal cells will just disappear and that's all. Again, in the same spirit, we have an expression for pk, which ensure positivity, so that's unconditionally positive. So that's for the ODEs. That's what we will use in a close future. And then, well, for the PDE, since we are looking for measure solutions, we will not use the classical upwind scheme, but the upwind scheme for measures. That's roughly the same, but with a slightly different interpretation that I will give you. So, first of all, we discretized the space for radius, as usual. Have a time discretization. That's there is no new things. There we have an expression for the velocity in that point. Well, that's not exactly what we used. We used the velocity at the edge of the cells of the mesh. But for sake of simplicity, here I will write the scheme at the center of the cells. So at each point rj, the measure a in at time t exponent k has approximately the weight agk. And so we will approximate the measure a at time tk by a sum of the weighted sum of Dirac masses. And in that setting, we obtain this numerical scheme, which is understood at to compute the mass at point G for the next time step. You just use this. You look at the flux, at the velocity, and the mass might move. Well, that is in the previous cell, and it moves to the cell of index j with a certain velocity. So that's basically a move of the masses from a point to another. With furthermore this term that to take into account the death of the adipocytes. And of course we have this line for the flux to take into account the birth of new adipocytes coming from the differentiation of pre-adipocytes. Here there should be a k plus 1 maybe. And that's all for the classical upwind. Well, the upwind scheme for measures. So here is the numerical analysis we made. Of course, proof of stability and convergence are yet to be made, but we will do this, we promise. And since we have this analysis, we will see some simulations.
Okay. So before beginning the simulations, we have to set the parameters. We have found the values of the parameters according to the literature and to some data that we have. So we did some data fit in order to get the parameters. With the current configurations, we have that both the mesenchymal cells and the preadipocytes have a proliferation rate that is smaller than the death rate and the proliferation rate and the differentiation rate. So we expect that either the mesenchymal cells or the preadipocytes will tend to 0. Moreover, we in a long time scale. In fact, we set the time scale to be 1 year in order to see what happens, how the phenomenon behaves for a large time scale. In these simulations, we have considered the velocity to be continuous, and these are the shapes of the proliferation rates for the mesenchymal cells and for the preadipocytes on the right. Okay. So this is the first simulations. In this slide, we can see the number of cells with respect of time. In the first plot, we have the trend of the mesenchymal cells. So what we see is that they tend to 0, as according to what we expected from the parameter setting. In the second, plot here, we see the preadipocytes. We have first a growth of the number of preadipocytes due to the fact that the mesenchymal cells differentiate into preadipocytes, but then they tend to 0 because they either die or they differentiate into adipocytes. This plot represents the number of adipocytes with respect to time. So we have an increasing of the number of adipocytes, and then we have a plateau. This is due to the fact that the death rate of the adipocytes is very small, and therefore with this time scale we can see the death of adipocytes. In the last image, that is this one, we can see the trend of the mean radius of the adipocytes with respect to time. And in particular, we see that at the beginning, so the initial configuration is that all the adipocytes have a minimal radius. We have a very fast growth of the adipocytes. In fact, the radius increases very fastly. Then we have a decreasing of the radius due to the fact that the preadipocytes differentiate into adipocytes. Therefore, we have a higher number of adipocytes at the minimal radius. And then we have again a growth of adipocytes that tend to the maximum radius. In this slide, we observe the distribution of adipocytes with respect to the radius. In the first image, we see the initial setting, that is the blue line. So we have that, as I said before, that all the adipocytes have a minimal radius. And we observe that they grow very fastly. This is due to the fact that the growth rate depends on the total surface of the adipocytes. In particular, is 1 over the total surface of adipocytes. So at the beginning, since the surface is small, because the radius of the adipocytes is small, we have a very large growth rate. And then, since they grow very fastly, we have a sharp drop of the velocity, because the surface increases. This last plot represents the adipocytes distribution at the last time step, so after 350 days. And here we observe that all the adipocytes are concentrated in correspondence to the maximum radius. And so we can see that this is a Dirac mass that is, and this is what we expected from the theoretical studies. Okay, and now we will speak about the spatial model. So we introduce the space into the model, and Pascal will present it.
Thank you. The spatial model is a mixture model in which we follow the concentration on mesenchymal cells, preadipocytes and adipocytes in space. So we need to introduce 2 new quantities, S of Tx, which is the volume fraction of the surrounding materials at time t and in x, and U of t and x, which is the spatial velocity of the species. So we get this system with in red the new terms. In this model, each each species move at the same velocity u. So we have three transport equations at velocity u with always a birth term, death term, and differentiation terms. And then we add also a special term on A. So now we need to say that there is no empty space in the system. So it will need to a closure condition that is given here. The sum of the volume fraction is equal to 1. And summing all the equation of the model, we end up with a constraint on U, that is a constraint on the divergence of U, which is equal to this quantity. And now we need to a way to define the velocity. So here we assume that U derives from a pressure Q through a Darcy law, this one, with an identity matrix as a diffusion matrix. And for the sake of simplicity, we take, when we take the divergence of this equation, we end up with a Laplace problem on Q. And for the sake of simplicity, we take homogeneous Dirichlet boundary conditions, but the biological relevance of this choice has to be discussed. And we also take non-incoming flux for M, P, and A. That means that cells can escape the domain, but no cells can enter from the outside. So now we'll present you some simulation with the spatial model.
Regarding the spatial model, we have applied the finite volume scheme in order to discretize the system. We first solve the Darcy's law, that is a Laplace problem. So according to the finite volume method, we have that this is the grid in, if we consider the 1D model, and this is an element if we consider a 2D model, so a square. For the transport system, we have applied an explicit upwind scheme, both to solve the spatial transport and to simulate the growth of the adipocytes. So I will show you the 1D simulations. It's important to notice that when we are in one dimension, the mesenchymal cells and the preadipocytes are in one dimension, while for the adipocytes we have to consider also the radius dimension. In fact, we have to take into account that the adipocytes grow in time. So this is the grid for the adipocytes, while for the mesenchymal cells and the preadipocytes we consider the interval that is the 0 and 1 centimeter. This is the initial configuration. So we have the mesenchymal cells and the preadipocytes are concentrated, are two Gaussian, concentrated in, centered in one point of the domain. While in this plot you can see the distribution of adipocytes with respect to the space, and that have a radius r. So in particular, we see that also in this case, all the adipocytes are concentrated in this point of the domain with respect to x, and they are all at the minimum radius. Okay. Now I will show some simulations. Okay. So in this plot, we can see the mesenchymal cells. So this is the first day, what happens in the first day. We have that the mesenchymal cells differentiate into preadipocytes. In fact, here you can see that the preadipocytes grow in correspondence to the concentration of mesenchymal cells. here we observe that the adipocytes number does not change in this small time scale, but we observe in this plot that the adipocytes grow very fastly. And in particular, we have that in correspondence of these 2 points, the growth is very fast. And this is due to the fact that, again, the velocity is, the growth rate is 1 over the surface at each point. And so in correspondence of these 2 points, we have a smaller number of adipocytes, so the surface is smaller and the velocity is higher. While here we have a higher concentration of adipocytes, then therefore the velocity, the growth rate is smaller. Here we see the mean radius of the adipocytes. So again, we see that here is smaller than in the other points of in space. And here we see the distribution of adipocytes with respect to the radius. So as in the 0D case, we observe that there is a formation of there is an accumulation of adipocytes in correspondence to the critical radius. Regarding the spatial velocity, actually we don't really see a spatial displacement because the spatial velocity is very small. And so this is a study that we are planning to do in future, in the near future. Okay. Okay, this is the okay. This is the last so the distribution in the last time step. So after 350 days, and in fact, again, we can see that the mesenchymal cells and the preadipocytes tend to 0. While the distribution of adipocytes, the number of adipocytes has grown with respect to time. We don't see the displacement again. We see that all the preadipocytes, all the adipocytes are concentrated in correspondence to the maximum radius, and so we can see it from this plot and also this one. So we see that all the adipocytes are concentrated here. Finally, we did some comparison with the experimental data. So in these 2 images, we can see here the mean radius with respect to time, and here the total number of adipocytes with respect to time. So first we observe that the 0D, the 1D and the 2D models give nearly the same results. So we can see that qualitatively the trends are similar. And moreover, we observe that especially the 0D model agrees with the experimental data. And this is due to the fact that the 0D model was calibrated with this data that we had. Okay, so now we will draw the conclusions.
Thank you. So, as a conclusion, I will emphasize some main observations that we made during this project. First, with the current setting of parameters, we see that the growth rate of adipocytes is very sensitive to the total surface of adipocytes. And it can be a problem because we have to choose very carefully the initial distribution of adipocytes in order to have a reasonable behavior of the model. So what we can observe also is that for the spatial models, we have always two very different time scales between the spatial displacement and the growth of adipocytes. And in particular, the spatial displacement is always very, very small. We also encounter some difficulties, for example, as I said, to choose the initial condition. And more generally, we have very few experimental data and too many parameters with respect to the experimental data. So therefore, a future perspective that could be interesting should be collect more data to better fit the parameter and initial conditions, and also set more biologically relevant boundary condition for the pressure, for example. So we could also investigate more deeply the stability of stationary solution, because, for example, for a wider range of parameters, and also see if we can say something theoretically about it. And finally, for the numerical scheme, it would be interesting to find a numerical scheme that is able to deal with discontinuous velocity, and also for the spatial parts, a numerical scheme that is able to deal with 2 time scales. Thank you for your attention. Okay, you mean for the direct mass, add another variable that... okay. In our first scheme, what we did is quite similar to that, because the last cell of the spatial domain has a non-outcoming flux condition, so it will store the direct mass, and on all the parts before these cells, we have a constant velocity in space, and that varies in time. And this.
We have the mean radius of the adipocytes. Actually, no, we don't know how they did the experiments, and also we are expecting more data, because it's very difficult to understand the real behavior of the adipocytes with this data that we have. So yeah, we are waiting for the data.
Well, so before going to lunch to feed our adipocytes, we would like to thank the organizing committee, which was not at all hostile, but show a lot of hospitality.